Good afternoon. How are you doing today? Hope everybody is well. Yeah? It's my pleasure to have Adrienne Kline today. Adrienne received her bachelor degree in engineering from Indiana Institute of Technology in 2014. In her bachelor thesis, Adrienne got interested in the brain-computer interface. After finishing her bachelor degree, she came to the University of Calgary to pursue a master degree with Dr. Janoronsky and Dr. Bradley Goodyear, and later fast-tracked into a PhD program. In her PhD research project, Adrienne is focused on studying functional MRI and electroencephalography, or EEG, as well as using image processing and machine learning for the development of brain-computer interfaces. Adrienne had her best scientific moment when she managed to simultaneously control two robot hands using real-time image brain activity data. One thing to point out, not only does Adrienne excel in school, she is also good in sports. A few years ago, she won a sports scholarship that allows her to play volleyball in the United States. Adrienne will defend her thesis next Tuesday and will start her medical school in July this year. Today, she will present her PhD work with the title Multi-Modal Imaging of Cortical Networks Controlling Lower Limb Locomotions Towards the Development of Brain-Computer Interfaces. Please help me to welcome.
Thank you, everyone. Quite the lengthy title. Multimodal imaging of the brain's functional connections during lower limb locomotion towards the development of brain-computer interfaces. First off, before I get sort of really started on my work so far, I'd like to sort of thank all of my supervisors, Jenna and Brad. I couldn't have done it without them, my research group, as well as my supervisory committee members, Dr. Syed and Dr. Forkert, and then my other examination committee members, because I'm using the presentation I'll be using on Tuesday, as well as all my funding sources, because without it, it wouldn't have been possible. So first I want to talk about sort of the motivation for this project, and it comes from sort of spinal cord injury. So spinal cord injury is kind of classified as a lesion in the spinal cord where you either have a complete severing of the spinal cord or a partial one. Depending on that, it can either stop the signal sending from the brain entirely or partially. And depending on where in the spinal cord that happens, the general rule of thumb is that it affects anything kind of below that lesion. So in both cases, whether you have paraplegia, sort of paralyzed partially or fully below the waist, your walking is impinged, as well as if you're quadriplegic, where you have sort of neck down paralysis and you also can't walk. So regardless of what kind of spinal cord injury, there's usually some sort of walking, mobility, locomotion affected. Also, with spinal cord injury, there's sort of two paradigms of thought about sort of solving that as a problem. One is stem cell therapies, sort of trying to bridge that connection in the spinal cord again to try to re-send that signal. However, this has had sort of minimal success. So the alternative now is sort of brain-computer interfaces, or BCIs, which actually try to bypass that lesion and then sort of reroute the kind of signals that are actually occurring in the brain to the muscles again. So with walking, there's a lot going on, as you guys know here at the HPL, that actually controls locomotion. So a big part of it is obviously sort of from the cerebrum, the brain, and then the other part is central pattern generators, which are sort of these automatic kind of movement patterns that we actually get just solely through our spinal cord. So, and these actually control sort of our flexors and our extensors, and then we receive feedback through joints, muscles, and skin, both sort of to our brain as well as our spinal cord. So there's several different technologies that we can actually use to kind of collect information from the brain. One is electroencephalography, or EEG, and it's a non-invasive brain imaging modality in which we have surface electrodes actually located on the person's skull. So this is a picture of a 64 electrode cap on a participant of mine. And it detects voltage fluctuations associated with neurons actually firing. So this is kind of an example of kind of the different readouts we would get. So EEG is usually attributed with different sort of spectral information, meaning we look at different frequencies. So there's a few different ones. There's beta, alpha, theta, and delta. The ones most highly associated with walking are alpha, beta, and gamma. That one's not on there. It's a little bit higher frequency. A little bit more on the EEG signal. So if our sort of little red cylinders represent our electrodes, we actually have, and this is sort of a blown up version of our neuron inside the brain, and this would kind of represent our sort of skin, meninges, scalp. What we actually have is a sort of a conduction through the neurons that actually then allow this voltage to be picked up. And it's because of this kind of neuron, a pyramidal neuron's orientation to the scalp. So this is the kind of neuron we're actually collecting from. So there's lots of neurons in the brain, but those aren't necessarily as responsible for the signal as these ones. And then the other technology I used was fMRI, or functional magnetic resonance imaging. So this one has the advantage that we actually have a little bit more spatial information. So with EEG, we had great temporal resolution up till about the millisecond level, but our spatial resolution was within about centimeters of one another, trying to actually figure out where in the brain that activity is occurring. So with fMRI, we have the inverse problem. We have very, very poor temporal resolution, but we have great spatial resolution. So we can actually see, as you kind of see here in the picture, the different sort of brain activities associated with this, and it can actually get quite deep inside the brain. Also to note that EEG, because it's a voltage fluctuation, it's a little bit more, it's more correlated with neuron activity because it's a direct measurement, whereas functional magnetic resonance imaging is a function that is sort of correlated with neuronal activity, meaning that it's a hemodynamic or metabolic. So it's not necessarily a direct measure. And how fMRI differs from normal magnetic resonance imaging, so I think sort of some people here might be familiar with it, is that, okay, you have a brain, but what makes sort of fMRI special? Well, it's actually the change in oxygen level to two different sites in the brain that are sort of demanding oxygen as a function of the fact that they sort of need it to be able to fire and function. So to continue with a bit of background information. So one of the goals of this project is to actually try to isolate individual right and left motor patterns, both with executed walking as well as imagined walking, because a person who has a spinal cord injury can only generate imagined signals. So one of the most difficult things with this is, sort of back to the spatial resolution idea, is if we kind of cut the brain, cut the brain in a coronal slice, as you kind of see here, then we get some around the motor cortex, then we get sort of a picture that's kind of mapped out like this. So this is kind of mapped out and known as a homunculus. So as you can see, kind of the parts that have been attributed to leg activity are very, very close together in that sort of brain fissure. So actually trying to separate those two becomes very, very difficult. Most of the EEG electrodes associated with sort of studying walking, whether it's actually on a treadmill, walking around a lab, or imagined signal stuff, is electrodes C1 and C2, which kind of correspond to those overlapping of that homunculus for those left and right motor areas. So that would kind of be here, which would sort of be the motor areas. And as you can see, there's not a lot of sort of spatial sort of differences there, because EEG is susceptible to sort of seeing the same signal, but just from slightly different positions. So onto my hypotheses. So I'll give you sort of a brief, so you don't actually have to read all that. And that is that I'm trying to combine sort of the two technologies, trying to capitalize on the fact that I have both spatial and temporal resolution of brain activity by using two technologies instead of just purely one. And towards the development of actually trying to isolate these left and right signals. So kind of onto the study. So I had 16 right-handed young healthy male participants, not spinal cord injured, because I'm also looking at investigating this kind of executed versus imagined classification stuff. So they were fitted with a 64 electrode EEG cap, as you kind of saw earlier. And then they were actually placed in the MRI with a device that kind of had to be custom built to both allow them to locomote in a supine position, and be able to collect dysfunctional magnetic resonance imaging. So you have a person lying down, and then they kind of have these pedals that actually kind of move back and forth, and it allows for both hip, knee, and ankle flexion. And associated with the timing of actual movement, so during the executed, we actually sort of have these MR response buttons incorporated, where it actually sort of timestamps the EEG activity, so we know where these stepping moments are happening, as well as it also incorporates simulated gravity because they're kind of lying down, so it tries to combat that. So my visual stimuli is, oh, it's starting already, great, it's a block design. So this means that it alternates between a fixation cross, which is what you're seeing right now. So this is kind of the sort of gold standard for baseline classification, and then it becomes sort of a CGI, computer-generated video image of sort of a person walking. And then essentially they're supposed to follow in time with that person, whether they're imagining or whether they're executing. So there's sort of two different sets of data collected for that. And so to sort of deal with the fact that you have head motion occurring while somebody's actually moving in the MRI, I had them walk at about 50 steps a minute, so it's about half the pace of normal walking. And so that would be kind of a limitation of the study, but and so this is just kind of a static version of kind of what you just saw. So you kind of have a baseline, and then it alternates between this for about a functional run of about 5 minutes in the MR scanner. And then this process is repeated for both executed and imagined cases. So onto kind of some of the post-processing as part of the study. So we have an MRI of a person's sort of brain and head, and we can actually isolate, based on just the artifacts that are generated by the EEG, where in space those are. So we can kind of say, okay, here's where the cap is exactly, and that way if it shifts during the data collection, or from when we put it on them to when we put them in the scanner, etc., we still have the classifications of those electrodes and where they are in space. So it kind of became about co-registering this EEG and fMRI information. So sort of bearing that, now we kind of have where EEG information is coming from, as well as kind of the distance with respect to fMRI. So what I sort of did, and this hasn't been done until this project, was to actually kind of come up with a proximity measurement for saying this is the value that that electrode is seeing for the entire sort of cluster of this kind of hemodynamic fMRI activity with respect to each EEG electrode. And this is kind of important based on distance, both from the actual sort of individual cluster to the electrode, as well as because within those clusters we have variation of values. So it takes into account both of those. Bearing that, what ended up working best, that slide's here, go by. So I ended up picking the top 4 electrodes that kind of came out of this proximity measurement. So that actually ended up being, for the imagined case, ended up being FC1, FCZ, FC5, and F3, not the typically studied C1 and C2. And so these kind of became the electrodes from which I then decided to try to actually isolate the individual right and left stepping to try to kind of come up with this classification algorithm. Sort of post-processing of the EEG data. So this would be for all 64 electrodes, but I'm only going to be kind of talking about the first 4 that I used. And so as per kind of this block diagram, what you had happening at each of those kind of top pieces of the block is a left and right alternation of the little avatar guy kind of walking. F, I kind of have labeled as the fixation cross, or the baseline to which this kind of set of data is actually compared. The reason for that is that otherwise it becomes kind of an absolute measurement rather than a relative one. So kind of chunking out that data associated with each left and right individual kind of chunk. I then filtered the data, re-referenced the data, and put it through a Fourier transform and actually summed over what the three frequency bands most commonly associated in EEG with locomotion, that being alpha, beta, and gamma. And so knowing that, I then sort of developed a machine learning algorithm. So now that everything is kind of in vector space. So we kind of have two sort of small sort of long vectors of information associated with each kind of cycle of walking over a number of participants. And we can then use this information to hopefully sort of data mine and actually find out individual differences between those right and left classifications. So the machine learning is a huge, huge field. But what I decided to do, because of the degree of correlation between the EEG signals happening, was to use neural networks as an alternative instead of something sort of quicker to train. So I'll just give you sort of a little quick demo, or not demo, a little explanation of how that works. So machine learning in neural networks tries to simulate a biological neuron in which we have sort of information input into the dendrite, and then we have some sort of all or none sort of threshold firing activity happening. And then depending on whether that activation is high enough, it conducts the signal towards the next neuron and sort of down the axon to subsequent things and carries out processes. So, and the same is true with an artificial neuron. So we have weights from either inputs or other previous artificial neurons in which we have weighting and inputs, and we sum those over a particular cost function. And then depending on whether or not that passes or not, we then follow up with subsequent information. So after many kind of humming and hawing and testing and retesting, my finalized neural network ended up looking something like this. So because I have 4 electrodes with 3 frequency bands in each one, I ended up having 12 predictors. So there's very few sort of general rules of thumb when it comes to network architecture, but the couple that there are is that you should have as many inputs as you have predictors. So I had 12, plus 1 more for sort of a bias component. So as per that, my input layer, or the green layer here, is my input layer. So this is where we're actually sort of throwing the data at the first sort of layer of this. I then, so this 2 blue layers here are now known as hidden layers. So this is where a lot of the information sort of processing, learning kind of takes place. So initially I actually had 3 hidden layers with 10 neurons in each, and then kind of tried to scale back until my classification started to drop. So I ended up finalizing with 2 hidden layers with 8 neurons in each, and for a classification output that was binary, i.e. right or left. And so the output layer was just simply 2. How do we actually sort of look at how well something classified? So we have a confusion matrix. We sort of have specificity and sensitivity. So we have predicted negative, or actual negative, actual positive, predicted positive, and sort of those type 1 or type 2 error type things. So here it's not necessarily true positive, but sort of, predicted left, but it was actually right. So, and the same would be true for that. So there's a couple of different ways you can actually train the algorithm. So you can train it over each person individually, or you can train it and throw all of the group data at it. So when I trained it as a group, it did quite poorly. So only at about 58.1%, so just barely above chance. Chance being 50% because it's a binary classification. When I did individual trained and then averaged across people, it became 92.3% for isolation of those 2. And then, then the classification, so then that's sort of the training portion of the machine learning, but then we actually have to test it. So we have to show it data that it hasn't seen before to see how well the network actually generalizes. So if it's predicted left, and the true left came out about 78.7, so using and for true right and predicted right, we had about 81.2. So using a chi-square test, this was actually came out significant. So meaning that using these predictors, we've actually been able to sort of isolate left and right with this degree of accuracy on a left and right foot basis. For an imagined case. So this is actually, so they're not actually moving in this particular case. Some limitations of the study is that I had 16 participants, and so while this is actually quite a reasonable size sample for fMRI, for solely EEG work, this isn't all that high, as well as all of mine were healthy right-handed males, so this kind of data doesn't necessarily generalize to older females that are left-handed. In addition, all of this was conducted in sort of a supine position, so it may be different if they were sitting up, standing, actually walking for the executed versus the supine condition, etc. The significance of this is obviously, so this is all happening offline, but the same sort of network architecture can be brought into real time relatively easily. And so for the brain-computer interface side of things, my emphasis has obviously sort of been on sort of the algorithmic signal processing side of things. But the end goal is to actually have some sort of effector that you have at the end with robotics, cursors, spellers, somebody who's locked in can now actually interact with their environment using prostheses, actually controlling a cursor on their computer to send an email, that kind of idea. Some future directions for this is that I want to follow up with, because there's so many different kinds of predictors and features you can use, you can mine this data for a very long time. And so the kind of idea of ranking these predictors so that you're actually trying to sort of come up with a balance between prediction speed and computational complexity. So that's important because if a person who has a spinal cord injury who's actually using a brain-computer interface is, sitting there and they think, oh, I want to, move my left leg with my exoskeleton, and your accuracy is 99%. That's great, but if it takes 5 or 10 seconds to actually compute that, it's too late already. So you want something that's very quick, but still has a high accuracy. So it's quite a balance between those two, and it very much comes kind of back to sort of the neuroscience, kind of picking features that actually predict this as well as we can with as few features as we can. As just kind of a little sort of proof of concept idea. Oh, I don't know why it switched, but I don't know why it switched. It was not sideways before. So sorry, you'll have to turn your head 90 degrees. But so this is actually with computer-generated imagined left and right signals. So this is kind of controlling actually a little sort of mini robot, as it were. So like I said, my sort of emphasis on the project was not on the robotic side, but more on the algorithmic side. But this is kind of an end proof of concept kind of thing. Thank you.
How many hidden layers did you use for your algorithm as you use what's it called? The learning?
The learning?
Yeah.
So I ended up finalizing with 2 hidden layers, but I started out with 3.
Why did you go back to 2?
Why did I go back to 2? So basically what it came down to was kind of changing these parameters one at a time and actually seeing what the training, how well the training accuracy did. So when I had 3, I think my highest training accuracy was about 88% on average, as opposed to 92, for example. And then so all of those hyperparameters kind of had to be developed one at a time, including kind of the learning rate, the number of neurons within each of those hidden layers. And so it's just kind of dictated by the classification accuracy on the training data.
Yeah. Why did you see the 4 best electrodes over like left premotor cortex and nothing so much on the right? Was the right close, or what was it with the left?
Yeah. So that was kind of dictated by the sort of algorithm I used for the proximity of the EEG with regards to the functional activity. So what ended up happening is you kind of say, okay, this is how far, these are the clusters within the brain. How far is this electrode from this cluster? And then you perform that for each individual voxel, volume pixel in the brain, and as well as the score that it's actually seeing, the Z-stat score that it's actually seeing. So it takes into account if the amplitude is higher or lower. So that wasn't something I selected. I developed the program, and then it kind of decided for itself. So that's not a machine learning thing. It was just a straightforward sort of computational program. And so the reason for the sort of CGI imagine conditions, selecting those became sort of biased off of those. However, I would say sort of if you wanted to go one step back and say, why is there more fMRI than happening perhaps on the left side of the lobe, on that side of the lobe? So out of the 16, 13 of my participants were actually right foot dominant. So that may be an indicator. I tried to use that as a predictor, left versus right dominant as a predictor. Unfortunately, it didn't necessarily show any promising results with that. The other thing that I found with some of the imagined condition stuff was more left lobe activation overall. There's also sort of there's also some association with language and sort of temporal timing of information. So I think that kind of had to do with why when a person's imagining, there's a bit more of this kind of, okay, I have to follow in line with this, and then there's word associations, even though I've tried to leave words out of it as much as I could.
So you said your individual classification rate was higher than your average?
Yeah, so it's kind of the difference of saying, here's 16 people with all of their data versus here's one person one at a time and then average the classification after that.
So does that mean every person has a different brain activation pattern for that specific task?
To some degree, yes. To some
And then
degree.
how would you implement that into your future directions where you say you want to have a person with a spinal cord injury being able to walk again if every person has different brain activity during walking?
Yeah, so I actually, it wasn't presented here, so in terms of sort of the repeatability and reliability between people, I did sort of a subset, not subset, just an analysis of the ability to differentiate between trials, because obviously there was about a whole bunch of different trials for left and right within the same functional run, as well as you have 16 participants across a couple different time points even. So what I found was about 20 to 25% of the variance was associated with people, whereas they were relatively consistent within themselves. Almost sort of 2% was about the maximum percentage of the variance attributed to within person variance. So that kind of means that I think we can train to a certain degree across all people, but that if the sort of telling message from that became, this has to be trained on an individual basis. So you can kind of have this, so this architecture works for all people, but that doesn't mean that, your data is going to work for me. Your training data will work for me and vice versa. So it means that we have to sort of train that information on a one-to-one user basis.
Thanks for the presentation. I have one maybe methodological question first. Like the one slide you show the brain activity across the channels.
Yes.
I was just curious, is that kind of a raw data trace that you would get, or is that already filtered? Because it looks like it's quite kind of low frequency.
Yeah, so this is already filtered, and this is sort of a more artistic rendition. So each of these blocks would be about 18 seconds long, but this window is probably about 1.
Oh, okay. Because okay.
Yeah.
That makes sense.
Yeah, so it's actually a lot sort of higher frequency, but because otherwise, to actually zoom out to sort of give you this kind of example here, you would be looking at a flat line.
Yeah. And then the other question I had is, so the end goal is kind of to record a brain activity from a person who maybe has a spinal cord injury, and that enables them to walk. Say, how would you transmit that signal to the muscles?
So there's a couple, so obviously this isn't sort of my project, but there's other people working on this. So there's a couple different ways. One is to actually have an exoskeleton that they wear. So there's some spinal cord injury people that just use it as a rehabilitation. And either they or their sort of therapist actually sort of has like a remote or something like that where they're saying, okay, left step, right step, left step, right step. So then it actuates for them. So there's other sort of motors and actuators sort of generating that kind of motion for them. So the exoskeleton is one option. I think it's at Berkeley, and they're doing functional electrical stimulation to actually bypass it. So they're using sort of the musculature and tendons and ligaments that are already there to sort of enact the same movement, but without all the extra hardware. So that's sort of two ways of actually sort of then sending that signal. So once you actually have that classification, sort of that's like solving one big problem, and then the other is kind of how do we solve this sort of yes or no signal now, and how do we correlate that to how much do we need to be sending to quadriceps versus hamstrings, and in what pattern to actually allow them to move.
Yeah, no, in that context, I was curious, at this point in time, wouldn't it just be enough to know whether the person wants to walk or not? Because most people want to walk with the left foot and then the right foot.
Yeah, exactly.
So is it even necessary to detect left and right, or would you just have to detect, well, this person wants to walk right now, so we're just going to activate left and right muscles to
Yeah, so I also did that as a separate classification, sort of active versus what would be rest, or sort of the baseline thing, and that accuracy was very, very high.
Yeah.
So I kind of looked at it like a flow chart, in that you sort of have information flowing in. Is it active or is it rest? If it's rest, then sort of terminate the process, or in future directions, it could be move my arm instead or something. But and then if it's sort of an active, then is it now left or is it now right?
So if I'm walking and 1 out of 4 steps is incorrect, that would be a huge problem. But you're getting 75% classification accuracy. So my question is there anyone who's working on error detection, linking errors between what your brain is seeing and what the output would be? And could there be like a kill process in there to end any incorrect movements that seem to be flagging?
I think I don't know of anyone specifically doing that, solving that problem exactly, but I would imagine that wouldn't be that much further of sort of a next step, because particularly with walking, where it should be sort of left, right, as opposed to sort of, like, oh, I want to move, my index finger versus that, like my pinky finger or something. The other thing that I know they're doing at Michigan is, University of Michigan, is they actually have a feedback system in which they sort of, the loop of the brain-computer interface kind of outline that I showed here. They actually have, they're actually trying to finish that connection now. So they're actually trying to give them sensory information back as well. So they say there's a paraplegic where they actually have him hooked up to a robotic arm, and they have sensors on the robotic arm, and then transmit the signal sort of the from the sensors there, and then actually transmit that back to the brain in sort of microstimulation, microvolts or something like that, so that they kind of can give that feedback to them, so they could almost error correct themselves in that sense too.
So I have a question.
Yeah.
So I finished looking at your presentation because I'm not in this field. I'm still wondering, like, why do you need functional MRI and EEG? So my guess is you need to train your network from the fMRI to connect with the EEG signal, so that you can use EEG signal to control your electroencephalography. Is that what it is?
Yeah, so you definitely could just take EEG solely. So I also did that as sort of a separate analysis. So this became kind of an fMRI-informed approach. So a lot of my thesis was based around differences in fMRI between executed and imagined, sort of just general brain differences, but then actually trying to use the information for the BCI, because fMRI has such bad temporal resolution, realistically and like financially and logistically, nobody's going to be able to walk around with an fMRI, or let alone pay for one, in a spinal cord injury kind of case scenario. And so it kind of became trying to capitalize on the information from the fMRI, but to sort of inform the EEG and feature selection from the electrodes. But I certainly could have just picked out different electrodes and said, I'm just going to use these. So
let me clear my mind. So you only need to do one scan of fMRI, and then use that to train your network, and then afterwards you can use just the EEG signals control.
So,
what do you mean by that? Is that every step I have to use an fMRI?
No, not necessarily, because fMRI doesn't even have the temporal resolution to tell you the difference between left and right, or at least not in the way I collected it, because I chose to collect it at 50 steps per minute. That's much too high a frequency for fMRI to actually try to differentiate the signals. I would have to slow the stepping down a lot. So the activation from the fMRI is kind of a combined signal from both the left and the right, so it becomes an on versus off signal, really, and where in the brain it's seeing that. So I'm not using the fMRI directly into the brain-computer interface. It becomes simply an electrode selection tool. So saying, this is where the hemodynamic or functional fMRI is happening. So based on kind of the likelihood that this is the electrode closest to this with this values, we're going to pick those. rather than actually trying to process fMRI data, which would be insanely computationally complex. So you're not actually using it yet.
Sure.
Seems like another approach would be to kind of do the opposite and try to optimize the way you could, like two different things you could think about to get your EEG signals as different as possible, so you could get your classification as maximal as possible. Have you tried?
I haven't tried that. So when you say sort of different,
Well, like
what
your
do
end
you have
goal
something
is,
in mind?
Your end goal is to try to maximize classification ability. Then you can optimize in the opposite direction. You just try and think about tennis in one case and think about reading Lord of the Rings. And presumably those two signals should be a lot different.
Yeah.
And then I can just drive whatever. I can drive something.
Yeah. So that would definitely, I mean, you could do that. The only problem is that I think for the better and better that these sort of brain-computer interface stuff gets, the more that we want to kind of keep different disciplines separate. So suppose now you have that same brain-computer interface. It's the same sort of kind of housing system and kind of computational system that you're using, but now they're doing a Google, it's hooked up to a, Google search engine, and they want to Google Lord of the Rings or something like that, right? Now you've taken that sort of data set away from something where you could have used it like that, I think. So I think the reason for that is trying to keep it as close to kind of what was before, what they like the signals associated with walking and stuff before, because in many spinal cord injured patients, they, obviously were kind of healthy, walking, normal people, so they know what it sort of used to feel like to do so as well. And so yeah, but in theory, yeah, you could do something like that.
What's the best paper on that shows that people with spinal cord injury can still imagine movements in a similar way to?
Yeah, so there's quite a few. So, most of the kind of information I've seen is that there's very little, the idea behind the spinal cord injury is that it's kind of just like you cut the connection rather than you actually have deterioration, like neurological deterioration like you would in Parkinson's, Lou Gehrig's disease, things like that. So kind of keeping with that in mind, there's not that much. There tends to be a slight degeneration of the actual spinal cord if you don't try to keep sending those signals. But as far as the kind of cerebrum cortex stuff goes, most of that stays intact. I know for some of the amputation studies that have happened where they've tried to brain-computer interface with the robotic hand because they've lost an arm, there is some remapping of motor cortex areas that happen with that. But that's a slightly different case because they no longer actually have that.
Yeah, Mike.
Okay.
The lady.
I'm just thinking of the next step where you do the brain-computer interface. I know in monkeys, a lot of times it takes them a bit of training and learning before they can use it. So I just wonder, if you take the fMRI data to first figure out which EEG electrodes to use, but then after a bit of learning and training, then maybe those underlying patterns might have changed to try and do this. Is it something that, do you think the fMRI is really going to, help with the end goal, or do you think you could have just stuck probably with, that EEG measurement and then using that signal to train whatever interface you need to do?
Yeah, I think you definitely could stick with the EEG, as well as there are some kind of ranking algorithmic things out there. The machine learning was kind of the last chapter of my dissertation, and so there's actually something called a relief algorithm. So you can actually rank predictors sort of within that. So I could say, here's 1,000 predictors, give me the approximate rankings and importance associated with that classification. So in theory, that would be an alternative to that. One of the things is, though, that takes a bit of time, isn't always that accurate, because the more predictors you have, the less accurate it is that that's the actual ranking within those predictors. As well as that varies on a person-to-person basis too. The fMRI kind of came from, so nobody's really done locomotion in fMRI, and the biggest reason for that is head motion. For the most part, is actually sort of, so you kind of saw my participants there, they kind of had to be strapped down. So one of the other sort of goals of the thesis was actually to try to say, okay, what are the actual structural, just fMRI brain differences between that? And then I said, okay, well, how could I use this to maybe help with my machine learning stuff? But you definitely could just do solely EEG and kind of brute force it too.
Yeah, because it almost seems like you can go the opposite way. Like if you have the brain-machine interface, get them to learn on it, then have the EEG with fMRI, and then from there make a hand selection of 2 or 3 that they would use in the everyday life outside the lab type. So.
Yeah, no, and you definitely could do that.
From my understanding, you use the fMRI data to select the electrodes you're going to put into your algorithm to
Yes.
train your machine learning. Did you, and you kind of explained that those could be confounded by language being localized to your left hemisphere. So did you try just like using the traditionally associated electrodes with walking, like C1 and C2, and just put that into your?
Yes, I actually did. So I used, so to kind of keep things fair, I used C1 and C2, as well as 2 other ones that were kind of frequently cited in the literature, PO3 and PO4, which are kind of parietal-occipital electrodes. And that classification was actually slightly higher than the fMRI selected alone. So that's actually sort of what was sort of an interesting sort of difference with that, is that with the fMRI there seemed to be sort of this temporal thing, whereas with the EEG you didn't necessarily have that as much. And there can be a lot of different reasons for that, and that's kind of comes back to fMRI is correlated with neuronal activity, whereas EEG is actually a direct measure of brain activity.
That leads into my question. Certainly fMRI measures brain activity, but neuronal activity can be stimulatory or inhibitory.
Yes.
Is that a bother in this?
It isn't with this. So all of mine, all of my bold signals are brain oxygen level dependent. So my fMRI signals were excitatory ones, not inhibitory.
How do you distinguish an active neuron that's inhibiting something from an active neuron that's stimulating something?
So back to that kind of, so it's done through a GLM, so general linear model, where I have, where is it? In my block design, where this sort of activation of shape is then convolved with the expected response, the activated response of that, and then it's kind of done as a how well does each voxel match that level of activation. And then so it becomes solely a measure of excitatory activation, not inhibitory activation.
So I guess I'm having, maybe we're miscommunicating, but
okay.
With something like a leg, you've got muscles on both sides of the joint, and they're probably co-contracting.
Yes.
And to make a movement, one of those muscles is going to have to be inhibited and the other one stimulated. That's going to come from neural activity, which is going to look like neural activity, whether it's stimulatory or inhibitory, to make it happen.
Yes.
Does that make sense? And you can't tell a difference in fMRI.
There is sort of kind of inhibitory kind of bold stuff, but I'm not as familiar with that and how that kind of plays out in sort of this sort of set of data, because all of mine kind of then become sort of scored on positive values, not negative values.
I might have missed it, but what was the result then for actual walking compared to imagined walking?
You're right that I didn't present it. So actual, the sort of the machine learning classification, or?
Yeah, the classification.
So I didn't run it for the machine learning classification, but I did some stats in a more traditional kind of statistics in a non-machine learning way, and I could still get relatively high prediction rates sort of with that.
Do you know if it's better or worse, or if it makes a difference?
It was actually, there were, for the same number of electrodes and the same number of predictors, the executed was actually worse than the imagined case. And that also kind of came back to that the imagined information seemed to be more, the imagined for the fMRI stuff was more diffuse and more extensive than for the executed. The executed was kind of localized to motor cortex and cerebellum, whereas in the imagined case you have motor cortex, supplementary motor cortex, you have some visual stuff happening, you have some temporal stuff happening, you have some frontal lobe stuff happening. So there's just a lot more activation kind of happening with the imagined signal anyway. So I think there was just a lot more sort of differences to be had between those two signals to start with. The filtering I did should have, because it was a relatively low stepping frequency, I initially thought, oh, maybe it's an artifact or something that's kind of skewing the data with the executed information. But because my walking frequency would have been cut off by sort of my filtering process, that shouldn't have been sort of a big issue. So I think it just had to do more with there's more activity happening in the imagining case, because I think, you know, how normal is it that a person sort of lies on a board and tries to imagine sort of walking using this sort of computer generated image. So oftentimes with more a task that's less usual, there's more activity because it doesn't know, your brain doesn't know how to do it efficiently yet.
Scott.
So your control, your neural net has two classes, right?
Mm-hmm.
Let's say quadriplegic, they want to move their arms and their legs.
Yeah.
Do you imagine building neural nets that have 4 classes, alpha classes, or having many neural nets that are all binary classifiers?
Mm-hmm. I think from a computation standpoint, I would say have multi-classes for at least kind of motor task type stuff, because I think if you kind of run the risk of kind of putting it through many sort of separate architectures rather than just one to start with, so long as you can get sort of the separability between those signals, which I imagine you could because I did, I would say that.
How would you imagine the complexity of the network architecture to grow with many more?
Yeah, yeah, because I mean, even with mine, I mean, I only used 12 of an available 192 predictors, and that was just for sort of the walking condition, right? So let's pretend I picked, 12 more for the, like for moving hands or something like that. Yeah, so now you'd have 24 inputs plus a bias layer. So you would go from 13 input neurons in the first layer to 25. And then I would expect probably, it's very hard to tell. So obviously at the end you'd have sort of 4 classes instead of 2. And then the in-between layer is very sort of test dependent. I would expect the architecture to be more complex, i.e. one more hidden layer probably, and probably a couple more hidden neurons within each of those layers. But I would imagine there would be some redundancy in those signals because they're both originating from the motor cortex. So I would think that you wouldn't necessarily need as many more neurons as you had predictors in at least the hidden layers. But I don't know that.
I have a question.
Yeah.
So nowadays the speech recognition technology is so good that do you think using brain signal is better to control the exoskeleton? It's better than using voice control.
Yeah.
Because like what Maurice said, well, you can just ask the exoskeleton to just walk, and you use the voice to control it. Walk, stop, go fast, go slow. Would it be better than using a brain signal since the brain signal is so complicated?
Yeah, I think that's actually a great idea. I think you could. I think you very easily could. And yeah, I don't know what the drawback of that would be, if any, but
Maybe someone else can control your exoskeleton.
yeah, I would say, yeah, if you have sort of mean friends maybe that, would be like, oh, start running. But so long as it, I mean, even, I know, like, I have a Google Home kind of thing, so it recognizes my voice, but it doesn't recognize, my friend's voice or something like that, most of the time. So you'd hope that would be true, and that kind of process would get better as well. So.
Different answer.
Yeah.
So during your protocol, you measured brain activity both during, like, when the person is actually imagining the task and when they're actually doing it, right?
Yes.
And so my question is, when they are imagining the task, do you have a lot more variability? Because I can imagine, like, if you actually do something, you don't necessarily think about it much. It's very kind of automated.
Yeah.
So it should be kind of a regular kind of signal. But if you're thinking about it, it requires a lot of focus to stay constant in your mind into, like, doing the motion and then thinking about that. Because if you have a case where the person already has a spinal cord injury, where they cannot do the task anymore and they just have to try to do it, but their body won't move, would they have that kind of variability, and would that impact the accuracy of your kind of learning algorithm?
If there did exist that variability, yes, it would. But the generalizability theory stuff I did in my thesis, which is kind of an extension of like just like correlation coefficient kind of information, is that it's actually kind of better to diagram it out, because if you have kind of all the variance sort of in your data, generalizability actually lets you parse out that data with effect to different facets. Or so I had trials. So how did people vary because they alternated within themselves? I had participants, so I had 16 of those, and I had 60 for each like left and right leg, and then I also had different time points. So now I can say, running this kind of paradigm, I can say this is the amount of variance associated with time points, this is the amount of variance associated with participants, so this is how much they varied between people, and this is how much they varied within trials. And during both executed and imagined, this was almost 0% in both executed and imagined conditions.
Okay.
Yeah. So that being said, they were the time points were only a week apart. So if a person actually did a lot of training, you did time points that were 6 months apart, that may very, very well change, right? I would expect that to change, but within a week, like my time points were very consistent. My trials were very consistent. It was my people that had a lot of variance between them. And that was kind of true for all of my conditions I tested.
Maurice, I guess I have one more question. This is just because I don't know much about the brain. What is it that makes you measure these different frequency bands in your EEG? And what is the origin of those frequencies in the brain?
Good question. So that's kind of a very, very good neuroscience question. And so I know for epilepsy, they do kind of these like big sort of phase type spikes as opposed to as much sort of spectral or frequency analysis stuff. But in almost all EEG literature, they're kind of alpha, beta, kind of gamma signals are attributed with different tasks. So alpha usually has to do kind of more with reflection and kind of a particular kind of thought, and then gamma is multisensory processing, and beta becomes more motor oriented. So where those signals originate actually sort of in the brain initially, I don't know. I just know that basically these are kind of these. I mean, EEG's been around for over 100 years, almost over 100, over 100 years now. So it's kind of a very established thing that these kind of frequencies are associated with these kinds of tasks. But why a particular frequency is, I couldn't tell you.
Based off of Maurice's question, do you think with your study or with your approach, you could have people do 2 different tasks within the same frequency band?
2 different tasks within the same frequency band.
So they don't only walk, but they also, I don't know, lift their hand?
Yeah.
Do you see that in your EEG signals, or?
Yeah, you probably would. So the frequency band stuff kind of comes from how much, so I Fourier transform the data. So then I have sort of a measure of how much activation you have like in alpha or something like that. So it would be interesting to see if you said, okay, just move a leg, how much information do you have now? Say, okay, move your hand and a leg. Does that information now go up or become less? So I didn't do that, but you certainly could.
Any other questions for Adrienne? Well, let's thank her again.
Now next week, please do join us again, because we have Dr. Jeremy Wong, who's sitting there. He's a research associate with Dr. Arkor, and he will explain to us sensory mechanisms for motor learning. I will see you next week.